Hello, my name is Lena Vincent. I'm a PhD candidate at the University of Wisconsin-Madison. And in this presentation, I'm going to share some of the work our research group has been doing in response to what's probably still the biggest obstacle facing agnostic biosignatures research. And that's our complete lack of understanding surrounding what the universal principles of life are. So our research group has been trying to understand life as a general phenomenon, and our general strategy has been to focus on the dynamic processes that are characteristic of life, meaning those that can really only be captured when a system is observed over time. And so we've developed both theoretical and experimental frameworks to try to explain how simple interacting systems of chemical cycles first self-organized into systems capable of key life processes like self-propagation and evolution, and how those could further complexify and eventually give rise to life. So we hope that this framework will help shed light on the universal principles of life, even as we don't know it, and therefore help advance agnostic biosignatures research. So I'm going to start with an assertion that I think most people will agree with, and that's that the ability to self-propagate and generate complexity through adaptive evolution are two essential features of life. And really, to be useful to agnostic biosignatures research, we have to try to explain if and how these processes can emerge independently of their biochemical constituents, including things like compartments, polymer-based genetics, and even specific metabolic pathways. Now, origins of life research has historically focused on trying to explain the origins of these specific features, but as a whole, they fail to explain how those features could emerge in the absence of a prior living process like evolution, and also how those very processes could emerge in systems that might look very different from modern biochemistry. So as an alternative, chemical ecology, which is the framework I'm going to focus on in this presentation, suggests that the processes characteristic of life might be able to emerge independently of these specific constituents, and is therefore much more likely to help us get at the universal principles of life that we're after in agnostic biosignatures research. So what is this chemical ecology model? So it starts with chemical ecosystems, and chemical ecosystems are basically sets of interacting autocatalytic cycles. And an autocatalytic cycle can be defined simply as a reaction or a sequence of reactions in which at least one of the chemical species involved is present both as a reactant and as a product, with more on the product side than the reactant side, and that's important. And we refer to these as member species or member chemicals, and they're denoted as M's or Mu's in the figures I'm going to show you. So importantly, these cycles also require food, or F, which is only present on the reactant side, and also generate waste, W, which is only present on the product side. Now, our research group, in an effort led by postdoc Zhen Peng, has been working to simulate autocatalytic cycle emergence of varying complexity, and found that the behavior of even the simplest autocatalytic cycles can be approximated using logistic growth models. And this basically just means that their behavior is very similar to that of biological populations. And even more interestingly, when there are sets of interacting chemical cycles that behave either as competitors, mutualists, or even predator and prey, then complex dynamics start to emerge in the system that resembles what you see in biological populations. And this can be seen in the concentration of member species over time. And you can see in these figures here that you see complex dynamics emerge. Now, a key consequence of this chemical ecology model is that it suggests that in slightly more complex ecosystems composed of more diverse autocatalytic chemical cycles, evolution might be possible as a net result of combination of different ecological interactions, in addition to any background random or stochastic processes that might emerge in the environment, like fluctuations in food species, for example. So our group has also been trying to explain how natural selection could take place in these systems under plausible prebiotic settings, like seafloors or geothermal pool environments, recognizing that a key component of these environments are mineral surfaces. And mineral surfaces have long been implicated in the origin of life because they do things like stick to or absorb organic compounds and also provide interesting catalytic functions. So chemical ecosystems that are stuck to or adsorbed to mineral surfaces, which we endearingly refer to as slimes or surface limited molecular ecosystems, could differentially colonize mineral surfaces as a result of their chemistry, and even mutate and change in colonizing ability as a result of rare side reactions. And the continual turnover of mineral surfaces might act to select for variants of slimes that are better at getting from grain to grain. And so this model shows how, in principle, you could get complexification and evolution in the complete absence of polymer-based genetics, or even compartments. So another important aspect of our research has been to try to test this chemical ecology framework empirically. And to do this, we've developed a new framework called chemical ecosystem selection, where we basically mix food-rich solutions with mineral grains and deploy an analog of experimental evolution. So to do this, we mix our ingredients together and give time for these putative slimes to emerge. And then we transfer a small subset of mineral grains to a new vessel containing fresh food and fresh mineral. And the idea there is to repeat this over many generations to selectively enrich for variants that are better able at getting from grain to grain. And different variants here are denoted as different colors. And so importantly, we also routinely sample both the bulk solution and the mineral surfaces to analyze them to track any changes that might arise in response to chemical ecosystem selection. Now, the nice thing about this procedure is that it's chemically agnostic, meaning that it doesn't presuppose any specific chemistry, and it can be deployed on virtually any set of conditions. The basic principle instead relies on detecting changes in proxy traits that arise in response to the selection procedure over time. So here's a figure showing hypothetical data to try to show how changes in a proxy trait that is related to self-propagation could change over time and reflect evidence of evolution. So this could manifest in a few ways. So we could have stepwise evolution in which the proxy trait and rate of propagation changes abruptly and is then maintained over subsequent generations. Or we could have a more progressive evolution where changes in proxy traits happen gradually in response to selection. Now, to give you specific examples of proxy traits we've been using to track slime evolution in our experiments, and to better illustrate the ecosystem selection approach, I'm going to give you a specific example of experiments we've run successfully in our lab. So our lab has screened several sets of conditions, but for the purposes of this presentation, I'm going to focus on one specific recipe that we used that gave us interesting results. But I just want to emphasize that in principle, ecosystem selection does not require this specific recipe. We only use this as a starting point, and our rationale here for the design was to try to replicate early Earth conditions as best we could. So we used a prebiotic soup that we modeled after spark discharge type experiments, the recipe for which you can find in this table. So you might notice that we also added additional compounds like ATP as a potential source of energy, but I just want to acknowledge that ATP is not a realistic prebiotic phosphate source, but we used it anyway. And on the mineral side, we used pyrite, which we ground up into powder and processed and cleaned ourselves. And we carried out all these reactions under anaerobic conditions. And in this experiment, we had 10 replicate lineages, which were independently propagated for 40 generations. And a generation in this experiment is 2 to 3 days. And we routinely analyzed these alongside controls, which only had one transfer in their history at the time of analysis. So we set those up strategically so that we'd be able to isolate the effect of transfers on any patterns that we saw arise. So one of the selected proxy traits that we monitored was the concentration of free inorganic phosphate, or orthophosphate, left in the solution at the end of a generation to approximate the amount of ATP hydrolysis that had occurred. And when we tracked this over generations, we saw this distinct oscillatory pattern, and you can see the tracking of each of those 10 independent lineages across generations there. And there are a few ways to interpret these oscillations, the simplest being that some kind of nonlinear or autocatalytic chemical system has arisen in our experiments. And when we compare this to what our theory group has shown, it's interesting to note that these types of oscillations are reflective of predator-prey type dynamics in interacting ecosystems. So this just hints at the fact that what we could be seeing reflected in these oscillations are the results of interacting autocatalytic cycles that have arisen in our experiments, although this needs much more validation to demonstrate conclusively. On the mineral side, we also observed distinct fractal structures on the mineral grains, and these were absent in control samples that only had one transfer in their history. And so these observations were done by scanning electron microscopy, and you can see representative micrographs here for an experimental sample that had 18 rounds of transfer versus our controls that had only one. And so we're still trying to figure out the composition of these fractal structures and determine if they're related at all to the oscillations we're seeing in the bulk solution. So it just further illustrates the fact that chemical ecosystem selection can be used to study complex spatial temporal dynamics that might arise in complex prebiotic mixtures. So while the results I've shown you are interesting and consistent with the appearance of interacting autocatalytic cycles, much more work is needed to determine whether these are true slimes by our definition. And to do this, we would really need to demonstrate evolvability. And so we plan on doing that by tracking these experiments for much longer, so at least for 100 generations, and carrying out more in-depth chemical characterization, which I'm going to get back to in a second. But on the theory side, our group is trying to understand the effects of spatial structure and heterogeneity on chemical ecosystem dynamics, the way might be imposed if we had mineral surface absorption. And the hope here is that this will help us better apply any of the findings from these theoretical experiments to our empirical experiments in the lab. And so a quick note on chemical characterization. So we plan on using untargeted chromatography mass spectrometry approaches to try to characterize any broad changes in product distribution that arise in response to this procedure, as opposed to focusing on just characterizing and identifying individual compounds, although we'd like to do that too. So we have a postdoc in our lab, Stephanie Colon Santos, who's been working on developing such methods for the characterization of complex mixtures, like the ones that you get out of the Foremost reaction, which are notoriously difficult to characterize and make sense of using more conventional methods. So we hope that by using these kind of untargeted approaches, we can see what changes arise as a result of the transfer procedure and how they correlate with the oscillations that we've observed, and just in general, help us get a sense of the chemistry underlying these putative slimes. So to conclude, we think that our chemical ecology model for the emergence of life will overall help advance our understanding of life as a general phenomenon by focusing on key life processes and how they can emerge in simple chemical systems. So I've shown you that chemical ecology and its framework can try to explain how chemical ecosystems of interacting autocatalytic cycles can yield complex dynamics that closely resemble what we see in biological populations, and how chemical ecosystem selection can be used to study such ecosystems under laboratory conditions. And as a bonus, that we've already found one set of conditions that yields promising results. And so further work in our laboratory will go to trying to understand these results a little bit better and further expanding both of our theoretical and empirical models to help better serve the community of astrobiologists trying to understand life as a general phenomenon. And finally, because the chemical ecosystem selection approach can be deployed on virtually any set of conditions, we might be able to use it to see whether conditions representative of other worlds could yield chemical ecology or lifelike dynamics, which will help us further assess the potential for life on other worlds, such as Mars or Titan and beyond. So with that, I'd just like to thank my co-authors, as well as members of our research group and our funding sources, and thank you for tuning in.